Today I'm going to give you an introduction to Chapter 4, a tour of the cell. There are two main types of cells, prokaryotic and eukaryotic cells. The domains of bacteria and archaea include prokaryotic cells, while eukaryotic cells fall under the domain eukarya and include organisms such as protists, fungi, plants, and animals. At the bottom here, you see two different images of prokaryotic cells, like the bacterium on the left, and then human uterine cells on the right, which are an example of a eukaryotic cell. And don't be misled by these diagrams. They appear to be the same size, but in actuality, the bacterium is a lot smaller than the eukaryotic animal cells. This diagram shows a typical prokaryotic cell, such as a bacterium. So bacterium is singular while bacteria is plural. And I'm going to point to a few things here. So some notable features is that all cells will contain DNA. So here is the single chromosome for the bacterium. And then the bacteria will also have the organelles that you see, these little dots. Those are the ribosomes. And then it is surrounded by a cell membrane and a cell wall. Now, some bacterium also have methods of locomotion or movement, and you can see this whip-like tail, these flagella, multiple of them to help bacteria be able to swim in a watery environment. So now that you've been introduced to prokaryotes, let's go ahead and compare prokaryotic versus eukaryotic cells. So prokaryote means before the karyon or kernel, so pro means early or before, and that kernel is referring to the nucleus itself. So eukaryotic cells do have a nucleus, so eu means true and karyon means kernel, so it does have a true kernel or nucleus. Prokaryotic cells do not have a nucleus. Their DNA is just coiled up. It's usually a circular chromosome in a nucleoid fashion. There is the fluid that all of this sits inside the cell called the cytosol or cytoplasm. And there are no other organelles other than ribosomes. Prokaryotic cells are fairly small and they tend to be more primitive, but they've been on the earth way longer than eukaryotic cells. So those are the first cells on earth, are the prokaryotic ones. And bacteria and archaea include prokaryotic cells. You may not have heard of archaeans before. They are also microscopic single-celled organisms that have some similarities to bacteria, but actually share a lot more similarities with eukaryotic cells, as we'll find out later. Okay, over under the eukaryotic column, true eukaryotic cells have a nucleus, which means that the DNA is inside, surrounded by a nuclear envelope. The fluid is also called the cytosol or cytoplasm. And now you're going to have specialized structures that are enclosed in membrane-bound organelles, and that allows the cell to be able to specialize certain functions within these organelles. Eukaryotic cells are much larger in size, they are more complex, they can be highly specialized, and so your typical plant and animal cells are definitely eukaryotic cells. At this point, I'd encourage you to pause the video and go to the link that's in Schoology for cell size and scale, and I want you to play along with this site to see different organelles and types of cells, to see their relative sizes all the way down to the atomic level. And when you're done playing with that, come back to this video at this point.
This graphic is really helpful to be able to see the relative sizes of a variety of things and whether you can see it with your unaided eye or with the help of a microscope. So the smallest thing that you could possibly see with your unaided eye would be a human egg, if you were able to view one. Larger would be frog eggs, chicken eggs, and then under the yellow shaded area, LM stands for light microscope. So we have these compound light microscopes at Longmont High, and you could see things at 40x all the way to 1,000 times magnification. But even then, you can only see the full cell. You can see the nucleus. Most bacteria, they're fairly small. And then the only real, besides the nucleus, the only real organelle you can see is the mitochondrion. Beyond there, smallest bacteria, viruses, proteins, lipids, all those are very small chemical compounds or items, and you're going to need EM, which is electron microscopy. And we do not have that at Longmont High. They're fairly expensive, and so you'd be using those at the college or professional level.
So most cells are pretty small, and that's because in order to be able to survive, to be really efficient at gas exchange, exchanging waste products and nutrients with the outside environment and moving materials inside a cell, cells have to be small to maintain a large surface area to volume ratio. And that large surface area allows cells at the cell membrane level to increase their rates of chemical exchange between the cell and the environment. So you can see here an example of some different size cells, and you've got the little cell with that one next to it. Its sides are like 1 by 1 by 1, and the larger cell is bigger. Each sides of this cuboid cell is 5 by 5 by 5. And then you see what happens if you break down that larger cell into smaller little cubes. And as you can see at the bottom, the surface to area ratio for the little cube is 6, and for the large cube is 1.2. So in terms of efficiency, that little cube is going to be moving materials through it at a faster rate and survive better than the larger cell or the larger cube right there. So now is a time to put your knowledge to work here. So which cell would be the most efficient at exchanging oxygen with the environment by diffusion? You should go ahead and pause this slideshow and then see what you would pick as the answer, and then resume again when you've picked your answer. Okay, I hope that you had picked that the cell with the largest surface area to volume ratio would be the most efficient, and it really doesn't matter what that shape is. Overall, what you are looking for is the largest number. The largest number here is 1.7, so this cube cellular shaped cell would have the largest surface area and would be most efficient at exchanging oxygen. Now, most cells in the body are not cuboid shaped. Most cells are spherical or round. So in order to calculate the volume of a sphere, you'll need to use this equation, V equals 4/3 pi r cubed. All right, let's take a look at this sample question. A certain human cell has a radius of 8 micrometers. Calculate the surface area, volume, and the surface area to volume ratio. To help you out, I've got some equations down here showing you the area or surface area of a sphere is 4 pi r squared, and the volume of a sphere would be 4/3 pi r cubed. Pause here, do your own math, see if you can come up with the surface area, the volume, and then the ratio between the two, and then continue on with this video to find the answer. All right, you can see here is my work, and the sphere radius is 8 micrometers. If you use the surface area equation, you're going to get 804 square micrometers. The volume, you're going to get 2144 cubic micrometers, and the surface area to volume ratio, you will take surface area divided by the volume, and you get 0.375. Did you get the same answers as I did? Now let's talk about a couple of real-life examples of why it's important to have increased surface area for cells to be able to have tissues and cells have the proper function, what they're meant to do. One great example for animals is taking a look at the small intestine here. So you can see that the intestinal wall has a lot of folds and wrinkles right here. So right there you're getting an increase in the surface area, so it's a highly folded surface. And if you zoom into a small section of the wall of the small intestine, you're going to see these finger-like projections protruding out. These projections are called villi. And then if you zoom into each villi and see the cells that are lining the villi, those cells lining it are epithelial cells, and they also have these extensions and projections at the surface of the cell called microvilli. So all of this is to increase that folding and surface area in multiple ways so that the main job of the small intestine, which is to absorb nutrients that you've digested, can be done very efficiently and effectively in your intestines. So as we take a look at this diagram, you're going to see that some of the nutrients that need to cross into the cells include triglycerides, fatty acids, monoglycerides, also proteins and sugars. All those, or actually not proteins, but more amino acids, they're going to be absorbed through the cell membrane itself. And so the more surface area there is, the increase in the amount of nutrients that the intestine is able to absorb. Of course, we don't want to forget about plants, so the main thing that plants need to absorb is water. Plants capture sunlight at the leaf surface, which also has a large surface area for solar energy collection there. But down in the roots, plants need to absorb water. So the roots are highly branched, you can see there in that diagram, and the surface of the roots are coated in these cells, also epithelial cells as well, and each of these cells have little extensions that come out called root hairs. And so again, you're increasing the surface area for absorption of water and minerals for plants. All right, from here on out, we're going to talk about the parts of a eukaryotic cell, and of course, eukaryote refers to the true kernel or the nucleus itself. So the function of the nucleus is that it is the control center of the cell. It contains the DNA, which is coiled up in chromosomes. You're also going to find mRNA or messenger RNA in the nucleus because that's where transcription occurs, where DNA is used to code into RNA. The nucleus is surrounded by a double membrane called the nuclear envelope, and the outside surface of that nuclear envelope is actually continuous with a rough ER or endoplasmic reticulum. Now, in order to get things in and out of the nucleus, proteins, RNA needs to travel through. So those little holes are called nuclear pores, and you can see them in the diagram on the bottom right here. These are pores, and they're kept open by proteins that surround the pores. The DNA is coiled up and condensed into a structure called chromatin, but it's not just DNA, it's actually DNA wound around proteins that are called histones. And as a cell gets ready to divide, this coiling increases to form visible structures called chromosomes. But for right now, in a normal cell that isn't dividing, it's that DNA material is called chromatin. Within the nucleus, you're going to see a darkened region called the nucleolus, and that is where ribosomal subunits are assembled. So ribosomal subunits are going to leave the nucleus to assemble together to form a functional ribosome to be able to do protein synthesis, but they are made and assembled, kind of the early manufacturing part happens in the nucleolus within the nucleus. Here's a great diagram of the nucleus. Let me point to a few key features. We have DNA wrapped around histone proteins. This is called chromatin. You see a darker region within the nucleus called the nucleolus. Here you see a double membrane. You can see that there are two layers of the nuclear envelope. Each of the pores is surrounded by proteins to keep those pores open. And then when you take a look at the DNA and zoom in on it, you will see that the DNA is formed into this mass called chromatin. The blue is the DNA, and the purple are the proteins that the DNA is wrapped around. So it's not just disorganized inside the nucleus, but the DNA is wrapped and coiled around these proteins. All right, let's move on to ribosomes. Now, ribosomes are found in both prokaryotes and eukaryotes because the most important job for them is to make proteins. So ribosomes are made up of rRNA or ribosomal RNA and proteins, and there's two parts to it, a larger part called the large subunit and the smaller part called the small subunit. So ribosomes, if they are floating freely within the cytosol of the eukaryotic cell, they are called free ribosomes, and generally the proteins that are produced from them are going to make products, proteins and enzymes that are going to be used inside the cell. Now, some cells need to make proteins for export, and so those ribosomes are going to be taken to the rough endoplasmic reticulum. They're going to dock there. The proteins that come off the ribosomal production line will actually go into the ER and then continue folding and processing in there. And generally, those proteins made in the ER are going to be exported from the cell. All right, now we come to your homework assignment. So you should find in Schoology or on the counter a handout that looks like a chart on cell organelles. And what I want you to do for the rest of this class and for your homework tonight is to utilize your textbook and Google and other resources that can tell you about cell and cell parts. You're going to fill out this chart. So for each item, like the cell membrane, do a brief little drawing or sketch, or if you're doing the digital version of this assignment, take a screenshot or photo and insert it right into that box. Use words to describe the structure of these items. List the main function or functions in the cell. And then on the last column, I want you to tell me, are these items found in plant cells, animal cells, or both? Have fun with this, and I hope you learn a lot about cell organelles here.